Hi, my name is Julie Thompson, and in this video, I'm going to be talking about DNA replication. So to get us started, I'm just going to give a brief overview. To understand DNA replication, you really have to know a little bit about DNA structure. So we'll actually start off talking about how DNA is built. And then we'll just get into some of the basics of replication, basic concepts before we get into the details of how it works. Next, we'll look at DNA polymerase, which is an enzyme that is involved in this process. And it's one of the main enzymes that helps to copy DNA. So we'll talk a little bit about that before getting into the real details of how leading and lagging strand replication occur. And then finally, I'll finish off with a little bit of summary of what we discussed. So before we get into DNA replication, I just want to take a moment to address what you're going to be doing in your task. And to demonstrate your understanding of this process, you'll need to make your own diagram of how DNA replication works. That diagram will need to include the list of items on this slide. And so I just want you to be really careful about the fact that you include all of these different items because if you miss one, it may be reason for your task to come back. So you want to make sure that you hit all of them, and I'll touch on all of these as we're going through the presentation. Okay, so how is DNA built? Well, DNA is a polymer of what are known as nucleotides. So a polymer is just a long chain that's built of smaller parts. And in this case, the smaller part is the nucleotide. And nucleotides themselves are built of smaller parts. So a nucleotide has three main parts. It's made up of a phosphate group, a 5-carbon sugar known as deoxyribose, and then it has a base. And in DNA, there are 4 different nucleotides, and what's different about each of the nucleotides is its base. And so we abbreviate the bases A, G, C, and T. So often when we're talking about DNA, we think of it as a string of A's, G's, C's, and T's. And in fact, for most of this talk, we're going to just be abbreviating our nucleotides with these boxes, right, of A's, G's, C's, and T's. Okay, so it's the order and the arrangement of these A's, G's, and C's, and T's that enable DNA to store information in cells. So just think about a short strand of 4 nucleotides like we have here. You could have quite a number of different arrangements of A's, G's, C's, and T's, each of which would specify different information. And in the human genome, there's 3 billion of these nucleotides. And so you can see there's a lot of different possibilities for how these can be arranged. Okay, so it turns out that DNA is not just a polymer of one strand of nucleotides, but it actually is a double-stranded molecule. And the nucleotides, they pair up with each other. So in this case, we've got G pairing with C and A pairing with T. And this is always the case. So these are called the base pairing rules. And in the double-stranded molecule, A is always going to pair with T, and G is always going to pair with C. The other thing that we want to take a look at with the structure of DNA is that there's different ends to the molecules. So if we look at this strand here on the left, it has a phosphate group at one end, and we call this the 5' end. And at the other end, it's got this free, it's actually a hydroxyl group, and this is the 3' end of the strand. If you look at how the strands are oriented relative to each other, they're flip-flopped. And so if we use our sort of pencil example, you know, the tip could be the 3' end, so that the tip over here is actually upside down relative to the other strand. So it's going to be like that, and we say that because of this arrangement, the DNA is anti-parallel. Okay, so we're going to be representing the DNA very simply, but be aware that it's a 3-dimensional structure, and it has what we call a double helix structure. And it sort of looks like a twisted ladder. So the 2 strands of nucleotides base pair together to form a double-stranded structure, and then that double-stranded structure sort of twisted into a helical shape. Okay, so for replication, it's kind of interesting that these base pairing rules, when the structure of DNA was discovered and we figured out that there's this base pairing that happens, the scientists that did that immediately recognized the implications that had for replication. So they hypothesized, and they were correct, that during DNA replication, what can happen is that these two strands separate or come apart, and that each of those old strands can be used as a template to make a new strand of DNA. And that's what happens. So the new strand is going to be shown in yellow here. So the T pairs with A, and then the A dictates that, hey, next we've got to put a T in, and so on, and the strand can be copied. And so once DNA gets copied, you have two molecules of DNA, each with one old strand and one new strand. Okay, so I mentioned at the beginning that one of the main enzymes that copies DNA is DNA polymerase. And specifically, there's a form of DNA polymerase called DNA polymerase 3 that's going to do the bulk of the copying. Okay, and so DNA polymerase 3 has some rules that it follows. So even though it's really good at making new DNA, it actually can't start the synthesis of a brand new strand on its own. It needs a little bit of help. And so its helper is known as primase. And primase can start the synthesis of a new strand. So what primase does is it's going to lay down a short segment of RNA nucleotides, which are slightly different in structure to DNA nucleotides. And what that does is it provides a starting point for DNA polymerase to work with. Okay, so the other kind of funny thing about DNA polymerase is that it can only make DNA in the 5' to 3' direction. So once it goes along, it has to be synthesizing 5' to 3'. It can't work in the opposite way. All right, so now we're going to kind of get into the more detailed points about how replication works. And we said that in order to copy DNA, one of the first things that has to happen is DNA strands need to separate. And that's the job of an enzyme known as DNA helicase. So DNA helicase comes along, and its job is to separate the two strands so that each can be used as a template. Okay, so again, we said that you're going to have to make a drawing of DNA replication, and so I want to kind of help you get started with that. We can kind of start with the helicase as our first step. So when you're drawing the DNA, you want to draw what's known as the replication fork. So I'm just drawing two strands. You can represent the DNA just as lines, which is simple. And you do want to label the ends of the DNA strand. So I'm going to label these 5' and 3', and then obviously the other strand would be in the other direction. Okay, and so what I need to do again is I need to represent helicase. So I would pick a symbol to represent each of your DNA strands. And so we can just use, for example, a green circle to represent helicase. And I want to make note that I use this symbol in a key at the bottom. I've got helicase, whoops. And you also want to provide just a short description about what helicase does during DNA replication. So helicase, it unwinds the DNA, and that's something that you could write. And then I'm showing that here, right, where I place DNA helicase at the replication fork, where it's separating the two DNA strands. The other thing I'll mention at this point is that for the DNA to remain separated like this, it's not very stable like that. So there's actually a second protein that comes along, and it sticks to the single-stranded DNA, and it holds it apart so that replication can happen. So I would, again, on my key, put a little dot and say that these stand for single-stranded binding proteins. Okay, so that just gives you a sense for how to get started with the replication diagram. Okay, so now we're going to get into the replication of the leading strand. And on the leading strand, things are pretty straightforward. And so the leading strand its synthesis is getting started right here. We would say that the template for the leading strand is here. And what I've shown with these blocks, this is the primer. So Primase has already laid down a nice primer for DNA polymerase III to start working with. Okay, so polymerase III is going to come in and it latches onto the 3' end of this primer, and now it can start adding DNA nucleotides. And it's working in the 5' to 3' direction, and the fork is also moving in this direction. Therefore, polymerase III is just going to be able to continue working and make one continuous strand on the leading strand side. Okay, so in terms of how can you represent this on your own diagram, again, you're going to have your replication fork, and you want to make sure that you label the ends of your DNA on all of your diagrams. And again, the leading strand is going to be down here. So what I would want to show, I would want to use something to represent my primer. And then it's probably good to use a different color to represent the new DNA. So I would just draw a line to represent my new DNA. And I would put an arrow here to indicate the direction of replication. And I'd also want to note that, again, this is the 5' end, so it's going 5 to 3. And then I need to pick a symbol to represent DNA polymerase III. So in this case, the blue circle stands for DNA Pol 3. Okay, so that's a pretty good representation of how to represent the leading strand and its replication. Okay, on the lagging strand, things are a little bit more complicated because our 5' end is here. The fork is moving in this direction, but replication with polymerase has to proceed in this direction. So what's going to happen is polymerase is going to come along and it's going to add DNA nucleotides to the 3' end of this primer, but it has to sort of say keep up with the replication fork. So all of the machinery that copies DNA is sort of complex together at the fork. And so this polymerase III, it can't get too far away from the fork. So what it does is it stops, it jumps back, and it starts again. So we'll add on to another primer at that site. Okay, so our representation of the lagging strand is going to be a little bit more challenging to show. Okay, so we said that we've got, and I'm just going to make this a little longer. And what I have to do is, because of the way that DNA polymerase III is sort of synthesizing some and then jumping back to the fork, it means that this strand is going to get made discontinuously in pieces. So I'm going to have to show multiple fragments. And I would say that the first thing that happens in the synthesis of a fragment is that an enzyme called primase is going to lay down an RNA primer, which I've shown in green. And then I'll show the primase enzyme with a green circle. So in this diagram, the green circle represents my primase. Okay, in the next step, in the second step, the primer's already there, and I want to show that DNA polymerase III comes along and it adds some DNA nucleotides. And again, it would be good to indicate the direction in which that happens. And again, we'll show our DNA polymerase III. We're going to show that with a blue circle. And so these are kind of the first steps in making these so-called Okazaki fragments on the lagging strand. Now, eventually, those fragments are going to have to be modified so that the primer's removed and that they get pieced together. And so we'll come back and kind of talk a little bit more about that. Okay, so what I've shown here is I've shown two Okazaki fragments. I've got one fragment here, and I've got a second fragment here. And each of these fragments has its primer shown in the maroon, and the new DNA is shown in yellow. Okay, so what's going to happen is there's going to be a polymerase that comes in and it's going to remove this primer. So that's kind of what we're going to see next. So this polymerase it's similar to the polymerase III, but it has a slightly different role, right? Because it's removing this primer and replacing it with DNA. So it actually is a separate enzyme. It's called DNA polymerase I, and kind of like Pac-Man, right? It's chewing up the primer and it's replacing it with DNA nucleotides. But what it can't do is it can't make this last connection. So you see that there's a space here. So this space needs to get addressed, right? And there's another enzyme called DNA ligase that comes in and it takes care of this. It seals the backbone of the DNA. So now this DNA is in a continuous piece. Okay, so when I represent my lagging strand, and before we had shown primase in step 1, and then we had another fragment that was still getting DNA added to it. And we also have to represent what's going on with these primers and their removal. So here I'm showing some Okazaki fragments that they still have their primers attached, right? So in sort of what I will call the third step, what needs to happen is we need to have DNA polymerase I latch onto the 3' end of this primer here, or this Okazaki fragment here, excuse me, and that is going to kind of bulldoze through the primer of the next Okazaki fragment, take it off. But when it gets to the end, it's got a problem, right? It's going to leave a gap like the one that I show here. So in our final step, ligase needs to seal this gap. So I will represent the ligase with a red square, and the ligase needs to seal these two Okazaki fragments. All right, so that's replication of the lagging strand. Okay, so this is an overview of the process. This is a diagram that I really like from a book called Campbell Biology. And on the top, you can see the leading strand getting replicated with DNA polymerase III. and it's moving along in this direction, and the fork is also moving in this direction. So again, that's going to get made in one continuous strand. On the lagging strand, right, things are getting made in pieces. We had these 4 different steps that we talked about on the lagging strand. That the first step in the synthesis of an Okazaki fragment is that primase has to lay down a primer. Once that primer is there, in step 2, DNA polymerase 3 is going to extend that primer from the 3' end and add DNA nucleotides. In the third step, DNA polymerase 1 is responsible for grabbing onto the 3' end of this fragment and removing the primer for the next fragment. And then finally, in the fourth step, DNA ligase comes along and it seals up the backbone of that lagging strand. So ultimately, we will get a continuous piece of DNA. The other things to notice, right, is that again in this diagram they're showing helicase that's responsible for separating the 2 DNA strands, and also single-stranded binding protein is shown at the fork, holding those two strands apart. All right. So I hope that this video was helpful in understanding DNA replication. If you have any questions, WGU students, feel free to send an email to biochemistry@wgu.edu. We'd love to help. Thanks.